Welcome back. Our next presentation is Cynata Therapeutics, and I'd like to welcome Kilian Kelly, CEO, to the stage.
Thank you, and good morning, everybody, and thanks for coming along to hear a bit more about Cynata Therapeutics. We're a clinical stage biotech company based in Melbourne, Australia, and we're focused on the development of an iPSC-based manufacturing platform, which we call Cymerus. Specifically, what we're doing with this is making iPSC-derived MSCs. I'd imagine everybody in the audience here is probably quite familiar with MSCs in general as a concept. Of course, these cells have been investigated very widely over the past 20 years or so for a pretty wide range of clinical conditions. And they've generated some really encouraging results, but it's fair to say there's also been some disappointments. And we think that a large part of that is due to the inconsistency in the cells when they're manufactured using conventional methods, which rely on an ongoing supply of new donors and extensive MSC culture expansion. I'll explain a little bit more in a moment about why we think they're problems. But that's really what the iPSC platform allows us to avoid. So by using iPSCs, we can make essentially all of the cells we'll ever need from a single blood donation, which of course we've made an iPS cell bank from. Now, the iPSC therapy field has really burgeoned in recent years, which is great, but we really consider ourselves a leader in this field. We completed the first ever iPSC-derived clinical trial worldwide. We've now got orphan designation and a cleared IND from the US FDA. We've generated compelling clinical data in graft versus host disease and diabetic foot ulcers. We now have 4 active clinical programs in 4 quite diverse conditions, and including a phase 2 and a phase 3 trial. And importantly, we're going to have 3 randomized controlled efficacy trials reading out roughly over the next 18 months. So just to expand a little on our pipeline. We've got a total of 4 programs, 2 of which are sponsored and funded by Cynata, and 2 of which are partnered and funded externally. So the Cynata-funded programs include graft versus host disease, which is currently in a global phase 2 trial with sites in the US, Australia, and Europe. Recruitment in that trial is ongoing. We're aiming to complete it within the next few months, and that should give us results from that trial in the second half of next calendar year. We also have a diabetic foot ulcer trial, which is actually now essentially completed from a patient visit perspective. The last patient visit was a couple of weeks ago. So we're now in the closeout stage of that trial, obviously analysing and aiming to release results in the next few months. We also have some initial data from that trial, which is very encouraging and which I'll share in a moment. Our 2 partnered programs include osteoarthritis, partnered with the University of Sydney and funded by the Australian government under an NH&MRC project grant. That's a phase 3 trial, randomised, placebo-controlled in 320 patients. We completed recruitment just under a year ago, but it's a 2-year follow-up on that trial because we're looking to show a disease-modifying benefit. So the final patient visit will be just over 1 year from now, with results a few months after that. Now, obviously, that's a particularly exciting trial because, as many of you will presumably know, there are currently no approved disease-modifying treatments for osteoarthritis. So we're aiming to be the first such treatment. The final clinical program is a trial that's just about to get underway, which is partnered with Leiden University Medical Center in the Netherlands. This is a trial where we're really aiming to use the MSCs as a replacement for immunosuppressant drugs, which of course are used to prevent transplant rejection. The group that is running this trial have previously conducted a similar trial using donor-derived MSCs with very promising results. So we're very excited about this one too, and this one is all ready to go. We expect the first patient to be enrolled imminently. So just to explain a bit more about our platform, again, I'm sure this audience is quite familiar with MSCs in general. They're really of interest because of their immunomodulatory properties and also their ability to promote tissue repair and regeneration. Importantly, they can be used in an allogeneic manner without matching the donor to the recipient. And they can also be engineered to introduce additional functional properties that they don't naturally have. But again, as I said earlier, the main problem or the main challenge in the field is how to make these cells consistently and at scale. And that's where the iPSC part of the story comes in. So again, I'm sure I don't have to explain to this audience what iPSCs are. The main advantage of iPSCs for us in this context is the fact that they have an effectively limitless proliferation capacity, which means we can produce essentially all of the cells we'll ever need from a single cell bank. The other part of the iPSC story, of course, is their ability to differentiate into any other cell type, which in our case is MSCs, and we have a proprietary process to do that, which I'll explain in a moment. But I really believe that iPSCs are the ideal starting material for the mass production of cell therapies. So in terms of how our process compares to a conventional MSC process, a conventional process relies on tissue donations, for example, bone marrow, then isolating MSCs from the donated tissue, and then the culture expansion process to generate enough cells. Now, the reality is a very extensive level of culture expansion is required. The reason for that is quite simple. You don't get many cells from an initial donation, perhaps 20,000 from a bone marrow donation, but you need a lot of cells for clinical use. So, for example, a typical intravenous dose in an adult is about 200 million cells. But of course, we're not just trying to treat one patient with one dose. We're trying to make large batches of allogeneic product, so an even greater level of culture expansion is required. Now, the reason that that's important and the reason that it's a problem is that MSCs, unlike iPSCs, MSCs do not have an ability to reproduce themselves to a limitless degree. So when they undergo extensive culture expansion, and this is very well established now, they undergo functional changes, then they lose potency and ultimately enter senescence. So that in itself is a problem. But to compound that, it means that there's a limit to how many doses can be produced from one donation, which means you've got to go back and get new donors all of the time. And the reason that's an issue is, again, it's very well established that there's a lot of variability in MSCs derived from different donors. So that just compounds the variability. And needless to say, when you're trying to make any sort of therapeutic product, it's essential that you can show that your product is consistent from batch to batch and over time. So that's really the challenge that we're trying to solve with our process. So by using iPSCs, as I said, they have an effectively limitless expansion potential. So we can make all of the cells that we'll ever need from a single cell bank derived from one donor, one time. And because we can get the scale at the iPSC stage of the process, when we then use our proprietary process to differentiate the iPSCs into MSCs, we don't need to go through a great deal of culture expansion at that stage. So we also avoid those problems that I mentioned when you do undergo extensive MSC culture expansion in terms of the functional changes, loss of potency and senescence and so on. So when you put all this together, what we have really is a way of making these cells with a high level of potency, consistency and scalability. Now, just to briefly summarize our clinical data so far, our first clinical trial was conducted in steroid-resistant graft versus host disease. This trial has actually now led to 2 publications in Nature Medicine, and the first of those was also featured on the front cover of that month's issue of Nature Medicine, which I think is a pretty unusual achievement for a small phase 1 trial. I think one of the reasons why it got such prominence was that it was the first completed clinical trial worldwide using iPSC-derived cells. But in addition to that, we also saw really encouraging results. So we treated a total of 15 patients with steroid-resistant GVHD, acute GVHD, I should clarify. 13 out of those 15 patients showed an improvement of at least 1 grade, which is considered to be an overall response. 8 out of 15 showed a complete response, and we saw a 2-year survival rate of 60%, which is really quite high in the context of this patient population, and I've got some figures in a moment to provide some context there. The other really important point is that we didn't see any safety concerns. So we had no serious adverse events or other safety concerns related to CYP001, which is what we call our iPSC-derived MSC product for intravenous infusion. So just to provide a bit of context here, this just shows the results that we saw in our phase 1 trial as compared to the results of a phase 3 trial with a drug called ruxolitinib, which is a JAK kinase inhibitor which is now approved in most markets for the treatment of steroid-resistant acute GVHD. And what we see is at day 28, the overall response rate with our product in our phase 1 trial was roughly similar to ruxolitinib in the phase 3, perhaps a little bit higher, but certainly higher than the best available therapy controls. And to clarify what that meant, it was essentially one of various other investigational agents for GVHD. But interestingly, what we saw then when you go out a bit further, for example, at the day 56 to 60 time point, is the response rate to ruxolitinib really dropped off, but that didn't happen with our product. And in fact, the response rate continued to improve. It was 73% at day 60, and then if we go out to day 100, it was actually higher again. And so there's a bigger difference there, of course, in response rates. And then when you look at survival, the difference is really quite pronounced. So as I said, we had a 60% survival rate after 2 years, whereas ruxolitinib had 38% after 18 months, and the best available therapy controls at 36% after 18 months. Now, the reason I'm comparing 2 years to 18 months is that the ruxolitinib trial, they didn't report 2-year survival. They said it was non-evaluable. Clearly, it can't have been any higher than what it was at 18 months. So I think there's a pretty clear suggestion there that we may be leading to a much higher survival rate. And the other important point then is safety. So as I've mentioned already, we had a very clean safety profile in our trial. That also holds true for the other trials that we've conducted so far, and indeed for MSCs in general. But unfortunately, that is not true for ruxolitinib, and it's not true for many other agents investigated for GVHD. So with ruxolitinib, for example, serious and life-threatening adverse reactions are quite common. They include infections of various types and various blood disorders and so on. So now moving on to our DFU, diabetic foot ulcer product. This is a novel MSC-seeded wound dressing, which is applied directly to the wounds, in this case for treatment of diabetic foot ulcers. We don't necessarily think its use is limited to diabetic foot ulcers. This is just the first clinical trial that we are conducting with this product. So the trial that's ongoing now, as I mentioned at the start, we've actually now finished the patient visits and we'll have final results within the next few months. But earlier this year, in around February, we released results from the first 8 patients per group after 10 weeks follow-up, and what we saw was really encouraging. So we saw a median reduction in wound surface area of about 88% in the active group, compared to 51% in the standard of care control group. So obviously, as I said, that's very encouraging, and we're optimistic that the results we'll see from the final data set, which is coming out shortly, we're optimistic that they will follow a similar pattern. So just to summarize the sort of catalysts that we have coming up at the moment, as I mentioned earlier, we have 3 randomized controlled efficacy trials reading out approximately over the next 18 months. The first will be diabetic foot ulcers within the next few months, followed by our phase 2 GVHD trial in the second half of next calendar year, and then followed by our phase 3 osteoarthritis trial early in 2026. Now, in addition to that, we'll have our renal transplant trial getting underway. We're expecting the first patient to be enrolled any day now. And the first cohort in that trial involves just 3 patients. It's initial, followed by a DSMB review. So we should have those initial results very shortly after the first patient is enrolled. So we really have a lot going on. And just to summarize, so we are focused entirely on our iPSC-based platform for the manufacture of MSCs. We believe this is a way to do that in a scalable and consistent manner. We believe that that's not really achievable using a conventional donor-derived process. We have a broad and rich clinical pipeline now with 4 diverse clinical indications. We've got compelling clinical data already from some of those, with a lot more to come over the next year and a half or so. And finally, I should mention, we are pursuing a partnership-driven business model, so we are seeking at the right time to out-license some of these clinical assets, and we're very happy to talk to anybody here who has an interest in finding out more about what we do and how we might work together. Thank you very much.